All right, welcome everyone. So we're going to begin a set of videos here looking at the second part of our immune system, that being our adaptive immune system, right? So if you haven't watched the first part, so immune system kind of part one, we talked about innate immunity, right? Our innate immune system. We talked about things like lines of defenses, first line of defense, second line of defense. We talked about various cell types, so macrophages, neutrophils, eosinophils, phagocytosis, interferons, inflammation, fever, things like that, right? This adaptive immunity lecture is now going to focus on the second part, and what we're going to focus on in particular, let me switch over here. What we're going to focus on in particular is the adaptive, or sometimes you may see it called the acquired immune system, right? So what this is going to involve specifically our T and B cells, okay? So we're going to look at T lymphocytes and B lymphocytes. It's going to be specific, all right? And you may also think of the term memory, right? So T and B cells is going to be specific, and there's going to be an immunological memory that is associated with this part of our immune system, right? So this term adaptive or acquired implies that we have to be exposed to something in order to gain an immunity to it, right, or towards it. And so we'll talk about later in this kind of part, we'll talk about things like vaccines, and we'll talk about T cells and B cells and different types of T cells and B cells. But we'll also talk about what's called a cell-mediated and antibody-mediated immunity, right? So one way to consider this is to look at a graphic like this, right? So this is showing adaptive immunity, right? So again, we're in our adaptive acquired immunity discussion. We'll talk about what's called cell-mediated and antibody-mediated, okay? So cell-mediated, this is occurring whenever there are pathogens, antigens, foreign, microbes that have entered cells. So they're within cells. And in order for our immune system to combat an immune response against it, it has to be presented to them, right? So in other words, the antigen, and we'll talk about the term antigen here in a moment, but the microbe, if you will, the virus, the bacteria, it has to be shown to the immune system. It has to be shown to an immune system cell, and then the immune system cell can then respond to it, right? So it's not that it's just recognizing it floating out in the circulation. It has to be presented to an immune system cell, right? So this is going to involve primarily our T cells, right? Primarily our T cells, and we'll talk about a few different types. The other division of this adaptive immunity is called the antibody-mediated immunity. You may also see it sometimes called the humoral immunity, right? Humoral immune system. And so the term humor is referring to fluid. And antibody-mediated means that this is going to involve antibodies circulating in our plasma, right? So circulating in the blood, that then bind to and attach to antigens, right? And so this is primarily going to involve our B cells, our B lymphocytes, right? But some cells, like our T helper cell, are actually going to be kind of an intermediate in between. They're involved in both the cell-mediated and the antibody-mediated, right? So a term that we need to understand is this one of antigen, right? So antigen, the term antigen means antibody generator, right? So anti in the antigen means antibody, and then gen means generator, right? So this is antibody generator. Now, what the antigen is you can think of it as a part of the pathogen, right? So in our context, we're going to talk primarily about foreign antigens, and we'll get to that in a moment. But think of it as essentially a portion of the microbe, the virus, the bacteria, right? So if you imagine, right, if we drew something, right? So if we drew like a bacteria, right? So if we drew a bacteria, right? Here's a flagellum. The antigens would be these little markers on the outside of the virus, or in this case, the bacteria, right? So depending on the source you look at, it may just refer to the whole thing as the antigen, but we're going to talk about a specific portion of the bacteria being the antigen. Okay, so we'll talk about foreign antigens primarily, but there's also self-antigens, right? And these are the proteins, the molecules that are on your own body's cells that your immune system recognizes as being normal, right? So your antigens, your self-antigens to you are normal, they're self, they're healthy. However, to me, your antigens would be foreign antigens, right? Because my antigens, the proteins on my cells, are different than the proteins on your cells, right? And so you have to consider this in the context of whose antigens are you referring to? Are you referring to ones that are innate within your own body, or are you referring to ones that are introduced to your body that are non-self, that are not healthy, right? Now, there's this other term called epitope, right? And so epitopes are essentially a particular part of an antigen, right? And so essentially think of it as just the most specific portion of the antigen that our immune system is actually going to bind to and recognize, right? And so we'll talk about in a little bit when we get to antibodies, we'll talk about how there's a specific region on the antibody called the antigen binding site, and it's binding to this very narrow specific region on the antigen called the epitope, right? There's other terms that you'll see, something called immunogen. This is an antigen that can induce an immune response, and if we are able to initiate an immune response, we have what's referred to as immunogenicity, right? So we have the ability to trigger a response, right? And really, it's the immunogenicity is from the perspective of the antigen, right? So it's immunogenic. It has the ability to trigger a response from our body, right? So through our T cells, through our various B cells. And then there's this other term called haptens that we're not really going to focus on, but these are small molecules that don't function as an antigen alone, but can if they are attached to some other molecule, right? So pollen, certain drugs, toxins in plants, things like that. Okay. So how do our cells actually bind to and recognize these antigens as being self normal or non-self foreign unhealthy, right? And so the way is through a receptor, right? And so recall, what is a receptor? A receptor is just a protein, right? So these are based on our DNA, okay? Remember, proteins are made from mRNA. mRNA is derived from DNA, which is our genetic code, right? And so we have all of these receptors on the outside of our cell. On the T cell, you call it a T cell receptor. On the B cell, you call it a B cell receptor. The B cells are going to directly contact antigens that are circulating, maybe in the plasma, in the fluid. And then T cells are typically going to have the antigens presented to them, right? And we'll have a whole discussion here in a little bit on what's called antigen presentation. Okay, we'll talk about this term called antigen presentation. There are specific cells that have antigen, or there are cells that are called antigen presenting cells, right? Now, if you think about it, there's millions of different types of antigens that we could potentially encounter, right? So think in our environment, in the world, in our community, there are hundreds of thousands and millions of different things that we could potentially encounter, right? So our cells have to be able to account for that wide variation, that wide variety in potential types of antigens. And remember, the antigens are proteins, or the receptors are proteins that are based on our mRNA, which is based on our DNA. And so you can have this process called recombinant shuffling, where parts of the genetic code get kind of shuffled and switched around, and that ends up producing a different type of receptor, so a different protein that has a different antigen specificity, right? And this idea of antigen specificity we will discuss and emphasize more in a little while, right? So this kind of graphic is showing our T cells along with their various surface markers, right? So you've got your various T cell receptors, right? So on helper T cells, on cytotoxic T cells, and then you've got your B cell receptors with their receptor, which are actually antibodies, right? And so we'll talk about the antibodies that are bound to B cells, and then we'll talk about antibodies that are freely circulating in the plasma later on, right? So we will have separate discussions on helper T cells, cytotoxic T cells, and B cells in a little bit. We're going to stop here. We're going to pick up in our next part, beginning to talk about this process of antigen presentation.